So thank you for this opportunity to allow me to discuss my research using induced pluripotent stem cells to identify pathways involved in the development of pulmonary fibrosis. Fibrotic lung disease is a chronic respiratory condition characterized by excessive collagen deposition and parenchymal scarring, leading to impaired gas exchange, as shown here on the picture to your right. Unfortunately, it can lead to respiratory failure in both children and adults. The current models include the bleomycin mouse model and, unfortunately, are inadequate in representing the progressive nature of the disease. The exact mechanisms leading to fibrosis are largely unknown, and this provides the basis for our investigation. Usual interstitial pneumonia, or UIP, is one type of fibrotic lung disease, and it is characterized by fibroblastic foci on histology, as shown here in the picture on the right. And these are thought to be collections of myofibroblasts that are alpha smooth muscle actin staining. UIP can be seen in the adult population in idiopathic pulmonary fibrosis and in the pediatric population in connective tissue disease and hypersensitivity pneumonitis. Human-induced pluripotent stem cells are a Nobel Prize-winning technology and have emerged as an important tool in understanding human disease. The principle is that adult somatic cells are taken from tissue biopsy and are genetically reprogrammed to induce pluripotency. These induced pluripotent stem cells are essentially like embryonic stem cells in the sense that they can differentiate into any cell line, including cardiac myocytes, liver hepatocytes, and any of the various types of lung cells. These differentiated cells can then be used for disease modeling, such as in Duchenne's muscular dystrophy, as we saw earlier today, and also in pulmonary fibrosis, which I will talk about now. These cells can also be used for drug discovery and cell-based therapy. Our objective is to develop a fibrotic disease model using human-induced pluripotent stem cell technology to better understand the pathophysiology of pulmonary fibrosis. My hypothesis is that pathways involved in fetal lung development are activated in our pulmonary fibrosis disease model compared to normal lung fibroblasts. We show that iPSC-derived mesenchymal cells, or fibroblast progenitors, produce a fibrotic phenotype disease model. We took primary fibroblasts from IPF lung tissue and cultured them in vitro. We did not see a phenotype at this point. However, when we reprogrammed into iPSC and then differentiated down a mesenchymal lineage or into a fibroblast-like state, we saw the emergence of these fibroblastic foci. These foci, as seen in UIP, are alpha smooth muscle actin staining, suggesting that they are myofibroblasts. Thus, we believe that reprogramming into iPSC and activation of these embryologic pathways provides a necessary stimulation for generation of this fibrotic phenotype. As a control, we took normal lung tissue and did the exact same thing and redifferentiated. However, we did not see this fibrotic phenotype as seen in this homogeneous appearance of the culture here. RNA sequencing showed that developmental pathways are predicted to be activated in our disease model. We looked at the gene expression profiling of our disease model and compared it to that of normal lung fibroblasts. As you can see in this heat map here, genes involved in embryonic morphogenesis were highly upregulated compared to our control. Additionally, we saw that the Notch pathway and Wnt pathway, and in additional data not shown here, that the TGF-beta and TNF pathways were also upregulated. So we already know that the Wnt pathway has been implicated in IPF, in pulmonary fibrosis. So this provides some additional validation for our disease model. To further investigate the Notch pathway and its contribution to pulmonary fibrosis, we did some exome sequencing, and we did see that there were single nucleotide polymorphisms that were predicted to be deleterious or disease-causing in the Notch gene in all 6 of our patients, thus lending more evidence to support the theory that the Notch pathway may contribute to pulmonary fibrosis. We conclude that reprogramming of diseased lung fibroblasts into iPSC provides a necessary stimulation to generate a fibrotic phenotype, suggesting the importance of reactivation of developmental pathways. RNA and exome sequencing demonstrate an association between PF patients and the TGF-beta, Wnt, TNF, and Notch pathways, which are known to be important for lung bud formation, epithelial differentiation, and branching morphogenesis. Thank you, and I'm happy to take any questions.
I am Mark Canada. I'm one of the pediatric nephrology fellows, and I'll be discussing fibroblast growth factor 23 and acute kidney injury. First, a little bit of background about fibroblast growth factor 23. It's a hormone that is secreted by osteocytes that induces phosphaturia and reduces levels of active vitamin D. Physiologically functioning as a phosphate and active vitamin D counter-regulatory hormone. FGF23 has major implications in chronic kidney disease. It's shown in the upper left-hand corner. FGF23 levels increase early in the CKD course before other mineral abnormalities and other mineral metabolism parameters are observed. As shown in the bottom left-hand corner, FGF23 levels increase as glomerular filtration rate declines. The table shows the ability of FGF23 to predict the progression of kidney disease. And finally, in the bottom right-hand corner, elevated levels of FGF23 have been associated with left ventricular hypertrophy, and the hormone has been shown to directly induce LVH, possibly providing a mechanism by which FGF23 contributes to CKD-associated cardiovascular morbidity. So CKD has been well characterized in CKD, but little is known about FGF23 in the setting of acute kidney injury. So our goals were to assess whether or not FGF23 levels are elevated in acute kidney injury and whether or not the levels can predict acute kidney injury. So to answer our questions, we studied the pediatric cardiac surgery population, which is well suited for the prospective study of acute kidney injury, as laboratory assessment is done immediately before and after surgery. There's a well-defined ischemic and or inflammatory insult to the kidney and cardiopulmonary bypass. There's a high incidence of cardiopulmonary bypass-associated AKI, and unlike the adult cardiac surgery population, relatively little comorbidity and polypharmacy that may confound assessment. So to date, we've enrolled 31 patients, 31 cardiopulmonary bypass patients in whom biochemical assessment has been performed before and after surgery, and of whom 20 developed acute kidney injury, which is defined as a 50% increase in your serum creatinine, and 11 did not. This table shows baseline characteristics. Most baseline characteristics did not differ between the AKI and non-AKI group, including the weight standard deviation score, height standard deviation score, cardiac complexity, cardiopulmonary bypass time, estimated GFR, and mineral metabolism parameters, including phosphate. The AKI group was younger than the non-AKI group, and as such had a lower creatinine than the non-AKI group. GFR did not differ. Yet, preoperative FGF23 levels were higher in the AKI group. And given that FGF23 does vary with age, we also calculated age-related FGF23 standard deviation scores, which we also observed to be higher in the AKI group. This bar graph shows the FGF23 levels over time at various time points post-reperfusion. FGF23 higher in the AKI group at baseline, and FGF23 increased in both groups within 2 hours post-reperfusion and remained elevated post-operatively. Post-operative values did not differ between the two groups. The ROC curve for AKI prediction showed that FGF23 standard deviation score tended to predict acute kidney injury with the area under the curve of 0.7. Now, we defined acute kidney injury per validated criteria, which defines acute kidney injury as a 1.5-fold increase in your serum creatinine. However, given the loss of power inherent to dichotomization and the fact that decreases in kidney function of less than 50% may still be significant, we also used a continuous outcome variable, that of percent change in GFR from baseline. And this shows the study sample. We excluded 2 patients that had GFR of less than 30. And of the remaining 29 patients, the median age was 30 months and the mean GFR was 110. And we found that after adjusting for age and cardiopulmonary bypass time, other factors that may affect the incidence of cardiopulmonary bypass-associated AKI, FGF23 standard deviation score still predicted % change in GFR from baseline. And we obtained similar results when we restricted our analysis to only those patients who had estimated GFR of greater than 90. Now, in assessing the predictive value of this hormone, FGF23, one question that arose is why are the FGF23 levels, which would be similar to those observed in moderate chronic kidney disease, so high to begin with? In a population with the average preoperative SDS score was greater than 2, in a population with normal renal function and relatively low phosphate levels is denoted by the FOS standard deviation score. So suggesting that there was another determinant of FGF23 in this study sample beside the renal function and the usual mineral metabolism parameters. So it actually recently has been shown that hypoxic rats have increased expression of FGF23. In our patients, we observed that those patients with a baseline oxygen saturation of less than 95% had higher FGF23 standard deviation score than those with a oxygen saturation of greater than 95%. FGF23 correlated negatively with baseline oxygen saturation, and even after adjusting for estimated GFR and phosphate standard deviation score, oxygen saturation still predicted FGF23 Z-score and did so better than the other 2 covariates. These data suggesting that hypoxia may have played some role or may play some role in the elevated FGF23 levels we observed at baseline in this population with otherwise normal renal function. Now, however, even after additional adjustment for oxygen saturation in our multiple linear regression analysis, preoperative FGF23 levels still predicted the development of post-op cardiopulmonary bypass-associated AKI. And that's it. I'll be happy to take any questions. Thank you.
Okay, so thank you very much for allowing me the opportunity to share my research with you today on targeted gene therapy and the treatment of X-linked hyper-IgM syndrome. The hyperimmunoglobulin M syndromes are a heterogeneous group of genetic disorders resulting in defects of immunoglobulin class switch recombination. So in order for a B cell to ultimately produce class switch immunoglobulin, it needs to interact with a T cell. And an absolute requirement is interaction between CD40, which is constitutively expressed on B cells, and CD40 ligand, which is tightly regulated and expressed only upon immune activation on T cells. Defects in CD40 ligand are what account for the X-linked form of disease, and these patients present with a combined immunodeficiency. Despite therapy with IVIG and prophylactic antibiotics, the long-term outcome for these patients remains relatively poor, with 20% survival at 25 years of age. Given these poor outcomes and knowing that all known disease-causing mutations are located on a single gene, this makes CD40 ligand defects an ideal candidate for gene therapy. There have been 2 previous studies looking at the role of gene therapy for XHIM using mouse models. In both studies, they were able to correct the immune defect. However, 2/3 of mice in the first study and 1/3 of mice in the second study developed abnormal lymphoproliferation, likely due to random integration of the cDNA gene using a retroviral vector. So these studies emphasize that gene transfer for genes that are tightly regulated can be dangerous and needs to be done in a more controlled fashion. So knowing that the CD40 ligand gene is tightly regulated and requires expression in its normal chromosomal context, we hypothesize that site-specific gene modification of the CD40 ligand gene in human hematopoietic or progenitor cells will correct XHIM. This can be done using site-specific endonucleases, which target specific DNA sequences for gene modification and ultimately allow physiologic expression of the corrected endogenous CD40 ligand gene. The endonuclease which we've been using are TALENs, which stand for transcription activator-like effector nucleases. They're a powerful genome editing platform derived from a plant pathogenic bacteria, and which can be manipulated in the lab to target specific DNA sequences. So the overall schema of the research would be to create talons that bind to the 5' untranslated region of the CD40 ligand gene. These talons would create double-stranded breaks, which can be repaired either through non-homologous end joining, which is error-prone, or if you utilize the sequences surrounding the talon cut site as homology arms, flanking normal human CD40 ligand cDNA, you could potentially allow homology-directed repair and overcome all downstream defects while maintaining control under the endogenous promoter. Thus far, we've designed and assembled several talon pairs that target the 5' UTR of the CD40 ligand gene. The best pair is shown here and has been able to achieve up to 31% gene disruption when introduced into K562 cells through electroporation. Now, moving forward with this talon pair, we created a GFP donor as a model for targeted gene addition in Jurkat cells. This GFP donor is flanked by homology arms that correlate with the talon cut site. This GFP donor is electroporated along with the talon plasmid pairs. And GFP expression can be assayed through flow cytometric analysis. As you can see here on the left, when cells are treated with just the GFP donor, we do not see any GFP expression, which is on the x-axis. However, on the right, when cells are treated with both talon and a GFP donor, we see the presence of a clearly GFP-positive population. When we track the expression of GFP over time, we see that its expression is consistent for weeks after electroporation, indicating that a permanent change in the genome has occurred. Now, since CD40 ligand expression is upregulated with immune activation, we would also expect that GFP expression would be upregulated with phytohemagglutinin stimulation. And so below we show that there is increasing CD40 ligand and GFP expression when cells are treated with increasing doses of PHA. These experiments indicate that the integrated GFP gene is under control of the endogenous CD40 ligand promoter. More recently, we've moved on to more clinically relevant cells. On the left, we have an immortalized T cell line derived from an XHIM patient that we follow here at UCLA and have been able to achieve up to 16.7% allelic disruption when treated with talon mRNA that we transcribe in vitro. On the right, we have normal CD34 cord blood-derived progenitor cells, and when treated with talon mRNA, can achieve up to 21% gene disruption. And so we have some very exciting, currently ongoing experiments in the lab, as we have a CD40 ligand cDNA donor cloned into a integrase-deficient lentivirus. And so we'll be evaluating for the reconstitution of CD40 ligand expression when patient-derived T cells and bone marrow CD34 cells are treated with both the talon and the cDNA donor. In addition, we have some recent experiments using the CRISPR-Cas9 system, which is another genome editing platform that has received a lot of attention in recent months, to target a patient-specific mutation, which I can share with you after this presentation. And so, of course, I have to thank my mentor, Dr. Cohn, for all of his support, my SOC committee, Dr. Garcia, Dr. Steem, and Dr. Crooks, and all of these people in the lab who work with me, who are much smarter than me and have gotten me this far. And of course, my funding sources, namely the T32 in human and molecular development, as well as the CDI training grant that has supported me for the majority of my last 2 years. Thank you very much.
Hi, everyone. So today I'm going to talk a little bit about our study, chlamydia and gonorrhea in HIV-infected pregnant women and infant HIV transmission. And I'd like to thank you for all the opportunity to present a brief summary of some of our primary research findings. So these are our disclosures. So in terms of a background, sexually transmitted infections such as chlamydia trachomatis and Neisseria gonorrhea pose a tremendous global health and economic burden, with approximately 106 million new infections of both chlamydia and also gonorrhea individually. In HIV-infected pregnant women in low and middle-income countries, they represent a particularly high-risk neglected group. In countries such as Brazil, as well as in South Africa, rates of chlamydia have been estimated to be between 4 to 10%, and for gonorrhea, approximately between 1 to 4%. These infections are important because they can cause serious consequences in pregnancy. Apart from the ability to potentially impact fertility, they can also lead to adverse pregnancy outcomes as well as adversely affect infants' health.
They also pose a potential risk for special complications in HIV-positive pregnant women. Prior studies, although they weren't specifically in pregnant women, have suggested that they may increase HIV viral shedding if untreated, and in addition, they also may have the possibility of enhancing HIV maternal-to-child transmission. So in terms of the background for our study was a secondary analysis of a parent study that was the NICHD HP10040 study. And that parent study was a randomized phase 3 trial that looked at mother and infant pairs that were from Brazil, Argentina, the United States, as well as South Africa. And that particular study really was focused on investigating 3 different infant antiretroviral regimens to prevent HIV vertical transmission to these infants that were born to HIV-infected women that didn't receive any antiretrovirals during pregnancy, so they were very high risk and were also not breastfeeding. The primary objective, though, of our study was really to look at rates of chlamydia and gonorrhea co-infection in this group of HIV-infected mothers. In terms of our secondary objectives that we wanted to look at particularly whether or not there were any predictors, such as geographic distribution or sociodemographics and biologic parameters that could help predict if these women would be positive for chlamydia or gonorrhea. And in addition, we wanted to look at whether or not these STIs had any relationship to being more at risk for adverse infant outcomes. And lastly, what we wanted to look at was whether or not these STIs had any impact on HIV transmission to infants. So in terms of some of the methods that we ended up using, we looked at the maternal urines that had been collected in the parent study that were collected at the time of labor and delivery, and they were frozen, stored, and batched, and eventually, when we were doing our study, we sent aliquots to Cepheid, which is in Sunnyvale, California, and they used their Gene Expert platform to look at chlamydia and gonorrhea through nucleic acid amplification testing there. So in terms of some of our primary findings that we found, we ended up looking at 1,373 urines, which was approximately about 82% of the parent cohort. And we found extremely high rates of both chlamydia and gonorrhea. So overall, the chlamydia rates in the mothers were approximately 18.1%, 4.6% for gonorrhea. Those that were co-infected with chlamydia and gonorrhea were about 2.5%, and overall in this cohort, approximately 1 out of 5 women were either positive for chlamydia, gonorrhea, or both chlamydia and gonorrhea. In terms of associated risk factors that we found in terms of the highlights, we found really high rates of reported prior preterm birth in this cohort in general, as well as really high rates of illegal substance usage, and both of those were associated with being gonorrhea positive. And in addition, we found that women that did not receive any prenatal care, as well as younger women, were also more likely to be infected with chlamydia and gonorrhea. And lastly, we ended up looking at infant outcomes with regards to these STIs, and we found that those that there was really not any significant differences in terms of the rates of sepsis, septic arthritis, congenital syphilis, and pneumonia among these different STI groups. However, we did find that mothers that were positive for gonorrhea at the time of labor and delivery were more likely to deliver preterm infants. So the main other thing that we ended up looking at last was really whether or not these STIs, such as chlamydia and gonorrhea, had any impact on HIV maternal-to-child transmission. So overall, in this cohort that we ended up looking at, the rates of HIV transmission overall were 8.5%. And those that were uninfected with any chlamydia or gonorrhea, the rate was 8.1%. However, when we looked at the chlamydia-positive mothers, the rates were 10.7%, so higher, and it was actually highest among infants that were co-infected with both chlamydia and gonorrhea at 14.3%. When we looked at this individually, we also found a modest association with chlamydia, so mothers that were chlamydia positive were 1.5 times more likely to transmit HIV to their infants. And when we in the data as well, because there was approximately a 6% difference between those that were unaffected and those that were co-infected with chlamydia and gonorrhea, it does suggest a possible association with those infections as well. However, because of the limited sample size, the p-values for this were not significant, and this is likely due to the power limitations due to our sample size, and we were unable to really detect significant differences with that group compared to uninfected for maternal-to-child transmission rates less than 17%. So in conclusion, our study found very high rates of both chlamydia and gonorrhea in this neglected group of HIV-infected pregnant women, In addition, we've found that these women are really high risk for co-infection with chlamydia and gonorrhea. Worldwide, in general, in most countries, particularly low and middle income countries around the world, they currently don't test regularly for chlamydia and gonorrhea in pregnancy. And so our data also makes a case for the need to really at least do some screening for these STIs at the first prenatal visit. And lastly, it does suggest as well that there might be some association with chlamydia and or gonorrhea with HIV, maternal to child transmission. However, further studies are really needed that are powered to detect these differences. So I'd like to thank my primary research advisors, which are Jeffrey Klausner in adult infectious diseases, as well as Dr. Nielsen, as well as my scientific oversight committee that also includes Dr. Bryson and all the people listed here. And we'd also like to thank all CEPHID for their support of this study, as well as the HP 1004 Study Network and the Impact Trials Network, and all the people that were involved in the studies and study sites and investigators that were involved with this study. Thank you.
Thank you for this opportunity to present my preliminary data on my research on the effects of macronutrient deficiency on brain development. So we know that acute uterine artery ligation alters the hippocampal structure at birth in rat IUGR offspring in both a region and gender-specific manner. However, the effects of macronutrient deficiency as encountered in maternal calorie restriction with IUGR has not been studied. Furthermore, the isolated effects of glucose transporter deficiency on brain structural development is unknown. Specifically, our lab questioned whether GLUT3 deficiency altered the structure of the brain development, and if so, was it an increase in cellular apoptosis? Was it due to an arrest of progenitor cell differentiation or something else? So our hypothesis is glucose deficiency, as seen in IUGR and GLUT3 heterozygous mice, interferes with neural progenitor pathways, leading to an abnormal developmental cellular profile, and that males will have more severe disease than their female counterparts. So for our study design, we used two mouse models that have been established in our lab. One is a late gestation maternal calorie restriction of 50%, and the other is a GLUT3 heterozygous mouse model, which we know has decreased glucose transport at the neuronal level. We performed immunohistochemical analysis in coronal brain sections at both fetal day 19 and postnatal day 10, and we used cell-specific markers to track the brain cellular profiles. Some of the neural markers that we used were nestin, that stands for neural progenitor cells, O2A and myelin basic protein for oligodendrocytic progenitors and mature cells, GFAP that marks astrocytes, and tubulin, which helps identify neurons. We employed dual staining with Texas Red and FITC along with DOPI for nuclear staining. Our plan is to quantify the expression of the proteins using ImaQuant software, and we plan to include about 6 to 8 samples per group, per age, per sex, with age and sex matched ad-lib-fed mice or wild-type littermates serving as controls. We will perform a standard analysis using a p-value of 0.05 for significance. And we expect that there will be a decrease in neurons and astrocytes with the calorie restriction murine model versus the ad-lib-fed mice at fetal day 19 and postnatal day 10. We expect to find similar findings in the neurons, in the GLUT3 heterozygous mice compared to the wild-type littermates. So these are just some preliminary findings, what I'm going to share with you. So on your left are two samples from controls, and these are calorie-restricted IUGR mouse models. These are all fetal day 19, and they are coronal brain sections. And the green marked by the yellow arrow is the GFAP that marks the astrocytes, and the white arrow points to the nestin, which marks the neural progenitor cells. And I hope you can appreciate that as compared to the control slides, the calorie-restricted IUGR has a decrease in GFAP staining or astrocytic presence. It appears that there is no difference in the nestin on both slides, but we will have to perform a quantification to confirm those. So we are going to also, after completing this, look at a graded calorie restriction model of 25% to compare that to the 50% model. And then what else is very exciting is that our lab has recently created an absent GLUT3 in the brain mouse model, and so we will go ahead and perform those studies on that as well. So in conclusion, it appears that late gestation macronutrient deficiency reduces the astrocyte lineage without affecting the neuronal lineage. These results may be related to the fact that astrocytic development occurs in late gestation, whereas neurogenesis is completed earlier on. These changes in cellular profile may underlie the neuropsychological presentation of IUGR, resulting in motor or cognitive dysfunction. I'd like to thank Dr. Shin and Dr. DeVascar, who act as my mentors, and my SOC committee. Take any questions.
So the title of our project is Incorporation of Vault Nanoparticle in Mouse Lung Epithelium. The general purpose of this study is to find a novel strategy to transport compounds and proteins of interest directly to the lung epithelium. So Vault nanoparticles were actually discovered here at UCLA in 1986 by Dr. Leonard Rome. It was initially found in rat liver cells. We have now known that Vault nanoparticles are actually ubiquitous intracellular nanoparticles that is found in multiple cell types. Although the function is unknown, the proposed functions have included nuclear cytoplasmic transport, mRNA localization, drug resistance, cell signaling, and nuclear pore assembly and innate immunity. So here in this diagram is a diagram of our nanoparticle itself. The reason why it's called a Vault nanoparticle is the highly conserved, high-arched structure that we see here. Actually, a nanoparticle is actually 2 voltamers. It's actually 2 parts. And 75% of the Vault nanoparticle is actually made by this protein called the major Vault protein, or MVP for short. And in the major Vault protein, what you see is that the N terminus, which is in the middle of the nanoparticle, or the waist of a nanoparticle, goes up to the C terminus. Within the N terminus, there's this domain called the interaction domain, or the INT domain. And the INT domain is important because that's how we're going to package our nanoparticles with our compounds or proteins of interest. The C domain, C terminus of our protein is also very important. And the reason for that is that our C terminus has a Z domain. And the Z domain is actually a domain that's a non-specific FC binding portion that we'll talk a little bit later after we talk a little bit more about the Vault particle. So within the Vault particle itself, it's 2 voltamers, and the 2 voltamers are composed of about 38 of these MVP proteins. The mass of the nanoparticles is approximately 13 megadaltons, which makes the Vault nanoparticle the largest ribonucleic protein described in eukaryotes. So targeting to lung epithelium, and this is what we're interested in the Londe lab. And when our Vault nanoparticles are complex with an anti-EGFR antibody, delivery can be increased by several fold. Again, the reason why we used anti-EGFR antibodies is that we know it's very abundant within the lung epithelium. So in this little diagram here, this little cartoon here, we have a Vault nanoparticle. And in naming Vault nanoparticles, we tend to name it from the N terminus to the C terminus. So the CP MVP Z Vault is CP for a cysteine-based protein, or cysteine protein that stabilizes our Vault nanoparticles. The MVP is the major Vault protein. The Z domain, again, toward the C terminus is that FC non-specific binding portion, FC binding portion. So when we complex it with any antibody, it doesn't have to be necessarily anti-EGFR, what happens is it binds to the close to the C terminus at the FC portion, allowing the binding portion of the antibody to remain intact. Once you incorporate that with a cell of choice, the antibody nanoparticle complex will bind to our cell surface receptor of interest, in this case, EGFR. The proposed mechanism why this is enhanced is that this stabilizes this interaction here and allows for more efficient endocytosis, which is the mechanism that is currently hypothesized of how these nanoparticles incorporate. So our hypothesis is that Vault nanoparticles incorporate into MLE15 cells, and their uptake is facilitated when bound to an anti-EGFR antibody. MLE15 cells being a mouse lung epithelium. So these are our results. So the incorporation of Vault nanoparticles is enhanced when coupled with an anti-EGFR antibody. And in this upper portion, it's just Vault alone. And what we did is we took MLE15 cells, here stained blue with DAPI stain, and we incubated it at body temperature, 37 degrees Celsius, with nanoparticles for 30 minutes. And once we did that, we can see that there's already incorporation of our Vault nanoparticles, that you start seeing aggregation around the cell surface. But when we actually complex our nanoparticles with our anti-EGFR antibody and allow that to incubate with our cells, our MLE15 cells, for 15 minutes at 37 degrees, this process is actually enhanced, and you can just tell qualitatively that that there is a more robust aggregation of our Vault nanoparticles. Now, and this is both at 20x, and when we actually increase it to 40x, and I hope you guys can see it, it's that you start seeing these little specks inside the actual, around the nucleus, and actually it looks like it's inside the cells. And those are what we propose to be endosomes that are incorporating in our cells. After we did this, though, we wanted to see, well, it looks like it looks like qualitatively that there's an increase, but we wanted to get some kind of semi-quantitative answer of why, how good or how robust of an increase we're getting. So what we did is we took J-image, and we did the fluorescent ratio of our red dye, which is in this case Texas red stain, and to a DAPI background. So what we see here is there's about a 3.5-fold significant increase in our incorporation of our nanoparticles when bound to an antibody. Interesting, and what we found preliminarily, and which is also interesting, is that the chemokine response is suppressed when coupled with an anti-EGFR antibody. And what we have here is just CXCL1, which is a chemokine, a neutrophil recruiter, and a fold induction, induction. And what we see here at the control being cells alone, there's not much of a response. And when we added the antibody and we checked for the qPCR for CXCL1, we didn't see much of a response either. But when we add our Vault alone, we actually see a significant response, actually about a 25-fold increase in our CXCL1 expression. Interestingly enough, though, when we add our Vault with our antibody, that huge, robust increase in CXCL1 actually drops down to almost baseline. That also was significant when compared to Vault alone. So in conclusion, Vault nanoparticles incorporate into EGFR-expressing MLE15 cells, and their uptake is enhanced when bound to an anti-EGFR antibody. The potential chemokine response due to nanoparticle exposure alone may be suppressed when coupled with an anti-EGFR antibody. So we'd like to acknowledge the Rome lab, in particular Valerie Kickhofer, who supplied us with the nanoparticles, Dr. Leonard Rome himself, who was very supportive during our study at the California Nanosystem Institute here at UCLA. And then we also like to thank the DeVascar lab, in particular Bolshu Shin, who helped us with our immunofluorescent pictures. Thank you very much.